Hi, my name is Sydney Eberth, and today's date is May 19th, 2020. And today I will be talking about the determination of gender based on genes and hormones. I will first begin this presentation by giving a little bit of background information on this topic. Humans are born with 23 pairs of chromosomes for a total of 46 chromosomes. 22 out of the 23 pairs of chromosomes are autosomes, which direct the development of the human body form and of variable characteristics like hair color and blood type. The last pair of chromosomes are sex chromosomes, and these are designated as either X or Y and contain genes that direct the development of internal and external sex organs. All cells of the body, excluding the sperm and the eggs, contain the 46 chromosomes, and these are known as diploid cells. The eggs and sperm are haploid cells, each containing a set of 23 chromosomes. When they come together to form a zygote, or a fertilized egg, they make a complete set of 46 chromosomes. One half of each chromosome comes from either the mother and the other from the father. Each male and female contain sex organs, which consist of the gonads, the internal genitalia, and the external genitalia. The gonads produce gametes, or the eggs and the sperm, and the male gonads are the testes, which produce the sperm. The female gonads are the ovaries, which produce eggs. The internal genitalia consist of accessory glands and ducts that connect the gonads with the external environment. Finally, the external genitalia include all of the external reproductive structures. The chromosomes that are inherited as a zygote will determine the genetic sex of the individual. Females are XX, while males are XY. The females inherit an X from each parent, and males inherit the X from their mother and the Y from their father. This means that each egg contains an X chromosome, while the sperm contains either an X chromosome or a Y chromosome. The Y is essential for the development of male reproductive organs. However, it is possible for chromosomes to be unevenly distributed. For example, if there is a zygote with the chromosomes XXY, that zygote will be male because of the presence of the Y chromosome. A zygote that only has a Y chromosome and no X will not survive because the X chromosome contains crucial genes that cannot be found in the Y chromosome. However, a zygote that has only one X and no Y will survive and develop into a female. However, two X chromosomes are needed to have a normal female reproductive functions. This is known as Turner syndrome. So, sex determination occurs around the 7th week of development. Before sex is determined, the embryonic tissues are considered bipotential because they cannot be morphologically identified as either male or female. The bipotential gonad has an outer cortex and inner medulla, which will either develop into testes or ovarian tissues based on a certain signal. The bipotential internal genitalia consist of two pairs of accessory ducts. These are the Wolffian ducts and Mullerian ducts. The bipotential external genitalia consist of a genital tubercle, urethral folds, urethral grooves, and labioscrotal swellings, which will develop into either male or female reproductive parts. The determination of sex depends on the presence or absence of the sex-determining region of the Y chromosome, also known as the SRY gene. And you can see the SRY gene over here in this diagram on the right-hand side of the screen. So if the SRY gene is present, the bipotential gonads will develop into testes. If the SRY gene is absent, the gonads develop into ovaries. In male embryonic development, the SRY gene produces a testis-determining factor protein, or TDF protein. This protein can bind to DNA and activate other genes whose products will direct the development of the testes. After the testes develop, they will secrete 3 hormones that influence the development of the male internal and external genitalia. Testicular Sertoli cells secrete anti-Müllerian hormone, or AMH. Testicular interstitial cells secrete the androgens testosterone and dihydrotestosterone, or DHT. Both testosterone and DHT are the major hormones in males. AMH causes the embryonic Müllerian ducts to regress. Testosterone converts the Wolffian ducts into male accessory structures, such as the vas deferens, epididymis, and seminal vesicle. Testosterone can also control the movement of the testes from the abdomen into the scrotal sac. DHT controls the remaining male sex characteristics, such as the differentiation of the external genitalia. In female embryonic development, the absence of the SRY gene signals to the bipotential gonad to develop into ovaries. With no AMH present, the Müllerian ducts will develop into the upper portion of the vagina, the uterus, and the fallopian tubes. With no testosterone present, the Wolffian ducts will degenerate, and without DHT, the external genitalia will develop into female characteristics. There has been a lot of research that has been done that indicates that female development is very complex and that multiple genes are actually required for the development of functional ovaries. Now, this slide depicts a simplified diagram that shows the progression of a sperm and egg coming together to form a zygote and then developing into either male or female reproductive parts based on the presence or absence of the SRY gene. And I would like to thank you for listening to my presentation on the determination of gender based on genes and hormones. Thank you.